Welcome to our exploration of comparative genomics, a fascinating field that examines the similarities and differences in the genetic material of different organisms. By comparing genomes, scientists can uncover evolutionary relationships, identify genes responsible for specific traits, and gain insights into the mechanisms of diseases. Comparative genomics has revolutionized our understanding of biology, providing a powerful toolkit for research in medicine, agriculture, and environmental science. In this video, we'll walk through the major applications of comparative genomics, highlighting how this field is shaping the future of science and technology. Let's begin by understanding what comparative genomics is and why it matters. Comparative genomics involves the analysis and comparison of genomes from different species. This approach helps researchers identify conserved sequences, gene families, and evolutionary changes across organisms. By aligning DNA sequences and examining genetic variations, scientists can pinpoint regions of similarity and divergence, shedding light on the evolutionary history of life on Earth. The field leverages advanced computational tools and massive genomic databases, making it possible to analyze vast amounts of genetic information efficiently. Understanding these foundational concepts sets the stage for appreciating the diverse applications of comparative genomics. One of the most significant applications of comparative genomics is tracing evolutionary relationships among species. By comparing genetic sequences, scientists can construct phylogenetic trees that illustrate how different organisms are related. These trees reveal common ancestors and divergence points, helping us understand the evolutionary pathways that have led to the diversity of life we see today. This information is crucial for fields like evolutionary biology, taxonomy, and conservation, as it provides a genetic roadmap of how species have adapted and evolved over millions of years. Comparative genomics allows researchers to identify conserved genes, those that remain relatively unchanged across different species. These genes often play essential roles in fundamental biological processes, such as cell division, metabolism, and development. By studying conserved genes, scientists can gain insights into the core functions necessary for life and understand how certain traits are maintained through evolution. This knowledge can also help in identifying potential targets for medical research and drug development. By comparing genomes, scientists can infer the function of unknown genes based on their similarity to known genes in other organisms. This process, known as functional annotation, is essential for interpreting newly sequenced genomes. If a gene in one species has a well-understood role and a similar gene is found in another species, researchers can hypothesize that the two genes perform similar functions. This approach accelerates the discovery of gene functions and helps build comprehensive genetic maps. Comparative genomics is a powerful tool for identifying genes associated with diseases. By comparing the genomes of healthy and affected individuals, or even different species, researchers can pinpoint genetic variations linked to specific conditions. This approach has led to the discovery of genes involved in cancer, genetic disorders, and infectious diseases. Understanding these genetic factors enables the development of targeted therapies and personalized medicine, improving patient outcomes and advancing healthcare. Agriculture, comparative genomics helps improve crop yields, disease resistance, and nutritional value. By comparing the genomes of different plant varieties, scientists can identify genes responsible for desirable traits, such as drought tolerance or pest resistance. This information guides breeding programs and genetic engineering efforts, leading to the development of more resilient and productive crops. The result is a more sustainable and efficient agricultural system that can better meet the needs of a growing global population. Comparative genomics also plays a crucial role in animal breeding and health management. By analyzing the genomes of livestock and companion animals, researchers can identify genetic markers linked to favorable traits, such as growth rate, milk production, or disease resistance. This knowledge supports selective breeding programs, ensuring healthier and more productive animals. Additionally, comparative genomics can help detect hereditary diseases early, allowing for better management and prevention strategies. Comparative genomics is extensively used in microbial genomics and biotechnology. By comparing the genomes of bacteria, fungi, and other microorganisms, scientists can discover genes responsible for antibiotic resistance, pathogenicity, or industrially valuable traits. This knowledge is vital for developing new antibiotics, improving fermentation processes, and engineering microbes for biofuel production. The ability to manipulate microbial genomes has far-reaching implications for medicine, industry, and environmental sustainability. Comparative genomics contributes to environmental science by helping researchers understand how organisms adapt to changing environments. By comparing the genomes of species living in different habitats, scientists can identify genetic adaptations that enable survival under extreme conditions, such as high salinity, temperature, or pressure. This information is valuable for conservation efforts, ecosystem management, and predicting the impacts of climate change on biodiversity. Comparative genomics has shed light on human evolution and migration patterns. By analyzing the genomes of modern humans, ancient hominins, and our closest relatives, such as chimpanzees and Neanderthals, scientists can trace the origins and movements of human populations. This research uncovers genetic changes that distinguish humans from other species and reveals how our ancestors adapted to diverse environments around the world. Personalized medicine relies heavily on comparative genomics. By comparing an individual's genome to reference genomes, doctors can identify genetic variations that influence drug response, disease risk, and treatment outcomes. This approach enables tailored healthcare strategies, ensuring that patients receive the most effective therapies based on their unique genetic makeup. Comparative genomics is thus paving the way for more precise and effective medical care. Cancer research has greatly benefited from comparative genomics. By comparing the genomes of cancerous and normal cells, scientists can identify mutations and genetic alterations that drive tumor growth. This information is crucial for developing targeted therapies, early detection methods, and personalized treatment plans. Comparative genomics also helps in understanding why certain cancers are more prevalent in specific populations, guiding public health strategy. Comparative genomics accelerates drug discovery by identifying conserved drug targets across species. By studying how different organisms respond to drugs, researchers can predict potential side effects and optimize drug design. This approach reduces the time and cost of bringing new medications to market, ultimately benefiting patients and healthcare systems worldwide. Comparative genomics is essential for understanding infectious diseases. By comparing the genomes of pathogens and their hosts, scientists can identify genes involved in infection, immunity, and resistance. This knowledge supports the development of vaccines, diagnostics, and treatments, helping to control outbreaks and protect public health. Rise of antibiotic resistance is a global health concern. Comparative genomics helps track the spread of resistance genes among bacteria, enabling scientists to monitor outbreaks and develop strategies to combat resistant strains. By understanding the genetic basis of resistance, researchers can design more effective antibiotics and stewardship programs. Vaccine development benefits from comparative genomics by identifying conserved antigens across pathogen strains. By targeting these shared components, scientists can create vaccines that provide broad protection against multiple variants. This approach is especially important for rapidly evolving viruses such as influenza and coronaviruses. Conservation biologists use comparative genomics to protect endangered species. By analyzing genetic diversity and population structure, scientists can develop strategies to maintain healthy populations and prevent extinction. This information guides breeding programs, habitat management, and reintroduction efforts, supporting global biodiversity. Synthetic biology leverages comparative genomics to design and construct new biological systems. By understanding how genes and pathways function in different organisms, scientists can engineer microbes to produce valuable compounds, such as medicines, biofuels, and materials. Comparative genomics provides the blueprint for building novel biological solutions to real-world challenges. Field of evolutionary developmental biology, or EvoDevo, uses comparative genomics to study how changes in gene regulation drive the development of different body plans and structures. By comparing the genomes of diverse organisms, researchers can identify genetic switches that control development, shedding light on the origins of complex traits and evolutionary innovations. Model organisms such as mice, fruit flies, and zebrafish are essential for genetic research. Comparative genomics allows scientists to translate findings from these models to humans by identifying conserved genes and pathways. This approach accelerates biomedical research and helps validate experimental results across species. Functional genomics seeks to understand how genes and non-coding regions contribute to phenotype. Comparative genomics provides context by highlighting conserved and divergent elements, guiding experiments that reveal gene function, regulation, and interaction networks. This synergy deepens our understanding of genome biology. Epigenetics studies heritable changes in gene expression that do not involve changes to the DNA sequence. Comparative genomics helps identify conserved epigenetic mechanisms across species, revealing how environmental factors influence gene regulation and phenotype. This research is crucial for understanding development, disease, and adaptation. Much of the genome does not code for proteins, but non-coding DNA plays important regulatory roles. Comparative genomics identifies conserved non-coding elements such as enhancers and silencers that control gene expression. Understanding these regions is vital for decoding the complexity of genetic regulation. Genome editing technologies such as CRISPR benefit from comparative genomics by identifying target sites and minimizing off-target effects. By comparing genomes, researchers can design precise editing strategies that are safe and effective, advancing gene therapy and biotechnology applications. Population genetics studies genetic variation within and between populations. Comparative genomics provides the tools to analyze large-scale genetic data, revealing patterns of migration, selection, and adaptation. This information is essential for understanding evolution, disease susceptibility, and conservation. Metagenomics involves sequencing genetic material from environmental samples. Comparative genomics helps identify and classify the diverse organisms present, revealing the structure and function of microbial communities. This approach is vital for studying ecosystems, human microbiomes, and environmental health. Phylogenomics combines phylogenetics and genomics to reconstruct evolutionary histories using genome-wide data. Comparative genomics provides the sequence information needed to build accurate phylogenetic trees, revealing the relationships among species and the timing of evolutionary events. Systems biology integrates data from genomics, transcriptomics, proteomics, and metabolomics to understand complex biological systems. Comparative genomics provides a framework for identifying conserved networks and pathways, enabling holistic analyses of cellular processes and disease mechanisms. Bioinformatics is essential for managing and analyzing the vast data generated by comparative genomics. Advanced algorithms and software tools enable researchers to align sequences, detect variants, and visualize genomic data. This computational power drives discoveries across all areas of genomics. Success of comparative genomics depends on open data sharing. Public databases such as GenBank and Ensemble provide access to thousands of genomes, enabling researchers worldwide to collaborate and accelerate discoveries. Data sharing fosters transparency, reproducibility, and innovation in genomics. As comparative genomics advances, ethical considerations become increasingly important. Issues such as privacy, consent, and the responsible use of genetic information must be addressed. Researchers and policymakers work together to ensure that genomic data is used ethically and benefits society as a whole. Education and outreach are vital for the continued growth of comparative genomics. By teaching students and the public about genomics, we inspire the next generation of scientists and promote informed decision-making. Educational programs and resources make genomics accessible to everyone. Comparative genomics informs public health by tracking the spread of pathogens, monitoring genetic risk factors, and guiding interventions. This approach enables rapid responses to emerging health threats and supports evidence-based policy decisions, ultimately protecting communities worldwide. Forensic science uses comparative genomics to identify individuals, determine relationships, and solve crimes. By analyzing genetic markers, forensic experts can match DNA samples to suspects, exonerate the innocent, and provide evidence in legal cases. Comparative genomics enhances the accuracy and reliability of forensic investigations. Archaeogenomics applies comparative genomics to ancient DNA, revealing the genetic history of extinct species and past human populations. This research uncovers migration patterns, domestication events, and evolutionary changes, enriching our understanding of history and prehistory. Pharmacogenomics uses comparative genomics to understand how genetic differences affect drug response. By identifying genetic variants that influence metabolism and efficacy, doctors can prescribe medications that are safer and more effective for individual patients, reducing adverse reactions. Rare diseases often have a genetic basis. Comparative genomics helps identify the mutations responsible for these conditions by comparing patient genomes to reference sequences. This approach accelerates diagnosis, informs treatment, and supports the development of new therapies for rare disorders. Plant pathology benefits from comparative genomics by identifying genes involved in disease resistance and susceptibility. By comparing the genomes of healthy and diseased plants, researchers can develop strategies to protect crops from pathogens, ensuring food security and agricultural sustainability. Marine biologists use comparative genomics to study the genetic diversity of ocean life. By analyzing the genomes of marine organisms, scientists can understand adaptation to extreme environments, track population changes, and discover new species, contributing to marine conservation and resource management. Food safety is enhanced by comparative genomics, which helps detect and trace foodborne pathogens. By comparing the genomes of bacteria from food samples and outbreaks, scientists can identify contamination sources and implement measures to prevent future incidents, protecting public health. Industrial biotechnology uses comparative genomics to optimize microbial strains for the production of chemicals, enzymes, and biofuels. By identifying genes that enhance productivity and stability, researchers can engineer microbes for efficient and sustainable industrial processes. Space biology explores how living organisms respond to the unique conditions of space. Comparative genomics helps identify genetic changes that occur during spaceflight, informing strategies to protect astronaut health and support long-term missions beyond Earth. Artificial intelligence, AI, is transforming comparative genomics by automating data analysis and pattern recognition. Machine learning algorithms can identify genetic associations, predict gene function, and accelerate discoveries, making genomics research faster and more efficient than ever before. Comparative genomics is a dynamic and rapidly evolving field with far-reaching applications in science, medicine, agriculture, and beyond. As technology advances and more genomes are sequenced, the potential for new discoveries continues to grow. By integrating comparative genomics with other disciplines, we can address some of the most pressing challenges facing humanity and unlock the secrets of life itself. Thank you for joining us on this journey through the applications of comparative genomics.